So yesterday in the earlier video I have discussed about how linkage is different from independent assortment. So today we'll be discussing about two concepts. One is the concept of pleiotropy and another one is the concept of polygenic inheritance. So first we'll be discussing about pleiotropy. Now what is pleiotropy? We have seen that one gene expresses one is responsible for expression of one character. Now it is always not that one gene will always express one character. There are cases where suppose there is one gene here in a chromosome, suppose gene X. This gene X is responsible for production of protein, suppose protein A, and this protein A is responsible for controlling multiple factors or characters. So this protein A is getting expressed in multiple characters. So that is the concept of pleiotropy. So let's say it is responsible for 1, 2 and 3 characters. So what is pleiotropy basically? Where one gene is responsible for more than one trait or one character. So that is called pleiotropy. Why it happens? Because as I said that a gene may be responsible for normally how the characters are expressed. Characters are expressed due to proteins which are expressed by the particular genes. Now when this gene will be producing such a protein, and that protein will be a common protein maybe for 3 different biochemical pathway, so that protein will be responsible for controlling all the 3 characters. So if there is by mutation means change in the genetic pattern, if by mutation the nature of this gene changes, so that will result in production of a defective protein. And since there will be production of a defective protein here, so all the 3 characters which were controlled by this protein or were dependent on this protein, there will be problem, or we can see defective character for all the 3 traits or all the 3 characters. Now this concept of pleiotropy was first observed by Mendel, where he observed that flower color was always associated. So what Mendel observed, that flower color of pea plants were always related with the color of the seed coat. So this was the initial idea of pleiotropy developed by Mendel. Now generally it is difficult to find out that one gene is responsible for how many characters. Now that becomes evident or clear when there is defect in the gene, because as I have already told you, the moment there will be defect in the gene, there will be defect in the protein, and when there will be defect in the protein, it will get expressed as a defect in the multiple characters which that particular gene is controlling. So here we can take the example of a disease which is called your sickle cell anemia. Now what happens in sickle cell anemia? Normally red blood cells are biconcave in nature or discoid. Due to sickle cell anemia, these red blood cells, they become sickle shaped in nature. Now this is due to a defective gene or defect in the beta globin gene. Beta globin is the protein present in red blood cells. So this protein is manufactured by a particular gene. When there will be defect in that gene, there will be defect in this protein, and due to defect in this protein, the cells will become sickle cell in. We will be discussing this further in the genetic disease part. Now, when this happens, it is also associated with other multiple defects, like there might be damage to heart, brain and lungs. So what we see that a defect in a particular gene is responsible for so many problems. One, the red blood cells are becoming defective. It is leading to brain damage to the brain. It is leading to damage to the lungs. It may lead to damage to the heart. So one defect in a particular gene is causing defect in multiple characters. So that is the concept of pleiotropy. Next we will discuss about the concept of polygenic inheritance. Now polygenic inheritance means, here we have seen one gene controlling multiple character. Polygenic inheritance means there will be multiple genes. Suppose in a chromosome there is gene A, B and C. So, or these genes may be located in different chromosomes as well. So all the three genes, they will be controlling a particular character, let's say X. The genes may be located, so don't make this mistake in understanding, the genes may be located in different chromosomes as well. So A, B, C genes are there, so they are all responsible for controlling a particular character. Now, polygenic inheritance is also called quantitative inheritance. Now, why it is called quantitative inheritance? If we take one example, we will understand it better. Let's take the example of human skin color. Now, human skin color, it has been found that it is controlled by three genes, which are indicated as A, B and C. So these are the dominant traits and these are the recessive alleles, A, B and C. Now it has been found that the skin color which will arise in human being is dependent on the amount of a pigment called melanin. More is the melanin, darker is the shade of the skin, and that melanin is produced by the dominant we can say dominant gene. So suppose the combination in the human being is capital A, capital A, capital B, capital B and capital C, capital C. That means the entire combination is homozygous dominant. So under such a condition there are 6 dominant traits or factors, so it will lead to maximum production of melanin and it gives rise to very dark skin tone. Similarly, if all the alleles are recessive, there is either no or minimum production of melanin. So under such a condition, no dominant trait is there, so I have written 0. So it leads to very light skin tone. Now if we take an intermediate condition, so there are only 3 dominant factors here, so it will give rise to an intermediate skin tone. So like that we can have different combinations. So what we find is that what kind of skin color will arise, that is a quantitative contribution of the 3 genes. These are 3 separate genes. This is one gene, this is one gene and this is one gene. So how all the 3 genes are contributing, depending on that we have specific skin coloration. So like that we may have only one dominant trait. Suppose it can be capital A small a and rest all are recessive. So this will give rise to, so it is 1, it will give rise to a light skin tone. So it is not that only A, it can be any one dominant So here it will also give rise like this, or it can be the C gene. So like that, so the overall expression of the skin color is depending on how many dominant factors are present from the 3 genes. So therefore it is called quantitative inheritance. Now we should understand one difference, that is the difference between polygenic inheritance and let's say what we learned earlier, multiple allelism. Multiple alleles. Now what is the basic difference? Multiple alleles, we discussed about the inheritance of human blood group. Now human blood group is determined by a single gene. The gene is a single gene, but it has different forms. It may be IA. It is found in 3 different alleles. The alleles are different. So these are the 3 alleles. So therefore it is a case of multiple allelism. The gene is one having 3 different forms. Here the genes are altogether different. 3 different genes are actually controlling a particular character, that is human skin color. So that is the difference between polygenic inheritance and multiple allelism. So we should not be confusing. So a quick recap, pleiotropy is where one gene is controlling more than one phenotypic character due to a common protein, whereas polygenic inheritance is multiple genes controlling a single character. So you go through what is there in the NCERT book before going, as you go through this video, you also read what is given in the NCERT book, the examples given there, and try to relate. In the next video, we'll be discussing about different genetic disorders.